Good evening. Welcome to the third season of Scientifically Speaking, an interactive online lecture series that connects world-renowned scientists to high school students and teachers. Carefully curated to inspire, excite, and ignite young minds in the field of scientific research, this interdisciplinary lecture series aims to help students develop a creative way of looking at life and connect their learnings to address real-world problems. Our speaker for the lecture today is Dr. Gitanjali Yadav, plant scientist at the University of Cambridge and NIGPR, Delhi. To moderate this session, we have with us Ms. Namrata Pandit from the Yuva School, Ranchi. Namrata is a science educator at Yuva School based in Hudub, Ranchi. She teaches biology and environmental sciences. She has a master's in life sciences, majoring in animal sciences, neurobiology, and ecology. She enjoys communicating science with varied audience of different age groups and often uses art as a medium. Her art for science is also featured in UNESCO's Creative Resilience exhibition this year. A very warm welcome to you, Namrata. Kindly take over from here and introduce our speaker for today.
Thank you, Parneet, for the introduction, and welcome everyone to today's session of Scientifically Speaking. Today, your speaker will be Dr. Gitanjali Yadav, and she, as already introduced by Parneet, is a plant scientist working at the Department of Plant Sciences, University of Cambridge, and also holds a joint appointment in India at the National Institute of Plant Genome Research, New Delhi. She comes from a diverse educational background with a PhD in microbial immunology from the National Institute of Immunology, New Delhi, a master's in biomedical research, and a graduate degree with honors in botany from the University of Delhi. Currently, she is a specialist in genomics and structural biology, with her work focusing on food security and plant communication. And Dr. Yadav has received several awards in recognition of her work. Some of them are the Hamid Fellowship from the University of Cambridge 2019, the Exceptional Talent Award from Royal Society of London 2017, the INSA Medal from the Indian National Science Academy 2011, and the Women's Excellence Award from the Ministry of Science and Technology, Government of India 2014. Now, I welcome Dr. Gitanjali Yadav to take over from here. Thank you.
Thank you, Namrata, for that nice introduction. And hello, students. I'm assuming we only have school children today listening to us. In case there are other researchers or somebody else out there, good evening. And thank you for joining the Scientifically Speaking series. I'm very happy to be presenting and talking to you today for this opportunity. So thanks to Ashoka. And I would like to begin by sharing my slides. Let's see. All righty, there. Oldest question on the planet. Can you see my slides? Okay, I hope you can. So once again, good evening and hello. I hope to be talking to you today about the phytochemical landscape, about the research that we do in the lab, and about what's happening out there that sparks our curiosity. And more than anything else, I look forward to hearing what makes you curious about things around you. If you just sit still in your garden, out in the yard, or close your eyes and think about nature, are there questions in your mind that you think you don't know the answer to, or that you think possibly nobody knows the answer to? I think that would be very interesting. And there are, of course, countless such questions. But for us to have those questions is where all of this curiosity and unraveling it begins. So I hope you can see on my slide, the dynamic mosaic of changing chemistry that we live in all the time around us is what we are trying to decipher in the lab. Plants now have evolved solely from the fact that plants cannot move and they mostly stay rooted to the spot. plants have evolved some of the most advanced forms of communication that we are aware of on Earth. And some of these forms of communication are what I'm going to be talking to you about today. Plants communicate with each other as well as with the rest of the biosphere through, yes, chemicals. And unpacking these chemicals, as you can see on my slide here, unpacking these chemicals is not just about pushing and merging the boundaries of modern technology and modern science, but it is also about being able to understand and appreciate how this chemistry is pivotal to human well-being. Phytochemicals are all around you. All of life as we know it is complex chemistry in a semi-permeable bag. Okay, but before I tell you what I'm doing in research, I have to tell you how we are doing these things and the tools that we are using to resolve the questions or the things that make us curious. Okay, mostly inferring plant communication and sometimes a little bit more. So when I say all of life is a complex chemistry in a semi-permeable bag, what do you understand from it? Those of you who are in India right now, if you've ever gone with your parents, trekking into the Himalayas or the Himalayan foothills, do you realize the sudden change that happens when you go into the Himalayas? There's a change, there's a familiar bluish tinge that completely covers the air. And that is monoterpenes. That's a specific type of chemical that is being emitted by pine trees, many others, but mostly pine trees. And so these monoterpenes in the air define how you see or think of the mountains or a visit to the mountains, and how it immediately puts in your mind the idea of refreshing, pleasant, cool, feelings, right? Also, if you're outside of India listening to me today, and if you've been to the south of France, you can smell the lavender or the scent of lavender even before you see it, because that scent is what you know that plant from or that color from, okay? Similarly, the scent of roses. Put in the chat box if you know something about different kinds of chemicals that potentially, or different kinds of scents that you associate with something or someone or some place and so on. And more often than not, it might turn out that it's a phytochemical that you're particularly thinking of. The scent of the rose, for example, or the yellow of sunflowers. It's not just the scents, but also the colors. So the yellow of sunflowers is also a kind of signal or a chemical message or a way of communication for plants. They have to constantly invite pollinators, right? And so bees and other insects have to recognize plants by their color, by their shape, by their texture, by their scents, and so on. And so all of this, including the way life gets transformed sometimes from this, from the way you perceive things chemically, is all about terpenes, as we call them chemically, but call them scientifically, or chemicals that are emitted from plants. Chemicals that are emitted from plants are phytochemicals. Okay, the scent of jasmine blossoms, the yellow of sunflowers, the flavor of cinnamon, taste, all that is there. It is all the different chemicals, and much of what you cannot see or fathom are part of a variable chemical code of plants. And that is the key word for our research, variability. I'm going to come back to that variability very soon. But before that, let's try and understand what you know or what you think about whether plants have anything or are anything more than vegetables. Does any of you think that plants can think? Well, in ancient scriptures, much of it, I will talk to you about what we know from recent research, but ancient scriptures carry information about plant life. Aristotle first said that plants have some kind of an inner life, including, he thought, thoughts, memories, dreams, and future plans. But then Charles Darwin, after his voyage on the Beagle, you know about Darwin, right? The guy who came up with the theory of evolution about natural selection. He went on the voyage of the Beagle. When he was a student, he was always failing in class. His dad was so sick and tired of him. Tera to beta kuch hone wala nahi hai. And he finally decided, okay, if you can't study sciences, if you can't study medicine, he tried the sciences, he tried the medicine, failed at everything. his dad said, okay, read scriptures, become religious, become a padri maybe, and that would be good for you. So he went to Cambridge to study, and there too, he just couldn't find anything of interest. And then Fitzroy said, I'm going for a few years trip. He was the captain of the ship, and he said, would you like to come along with me, young man? I could listen to a few things about God. And Charles Darwin took that opportunity and said, all right, I'll come with you. He was not a naturalist at that time. After the visit of the bee, after the voyage of the Beagle, when he brought back all of the different data sets and had seen much of the world, and before he came up with the idea of evolution, Charles Darwin, during that time, had also noted that plants do have an ability to communicate with the environment and translate this information into active movements of their organs. And this was way back in 1880, right? And that Charles Darwin thought about his idea of the evolution, and that was one of the greatest ideas human beings have ever had. And then what did he do with that idea? He put it into a drawer in his study table and shut it up for 20 years. 20 years later, another young student who had heard about him said, oh, I like your idea so much, and this is what I think is happening in evolution. And then Charles Darwin said, hello, that's my idea. I got it 20 years ago. And that's when he decided to publish it and make it known to the rest of the world, and the rest is history, okay? So let's stop with Charles Darwin there. Just a few decades after Darwin came an Indian scientist, J.C. Bose, who was a physicist actually, but he showed experimentally that plants do have cognitive abilities, okay? And that was 100 years ago, more than 100 years ago now. And he also showed it as part of an experiment that he live presented at the Royal Society at that time in 1903. And then, of course, Barbara McClintock, she thought plants were thoughtful. What do you think about it? Well, research over the last 100 years has shown that plants can indeed recognize other organisms and very complex interactions with birds and insects. They can entice pollinators. Don't go to the other plant. Come to me, please. My flower is dazzling, more pretty and larger and smells much better than what you're going towards. And then the bee or the insect will buzz around, come to the flower, to this flower. And also plants are often domesticating bacteria. So this is happening in all sorts of peas. The pea family, we call them legumes as scientists. And domestication of this rhizobium bacterium is happening by all legume plants in order to grab a certain critical nutrient that they cannot make by themselves, okay? And does anybody know what that critical nutrient is? Well, if you do, put it in the chat box. If you don't, I'll tell you at the end of this talk, okay? And of course, plants are constantly communicating with and rewarding a certain kind of insect for their own protection. And they are also, how should I put this? Performing agriculture of their own kind with ants. So they are constantly I shouldn't say hiring, but engaging ants and then rewarding them for working with them. Okay, and plants are also recruiting enemies to herbivores. If there is a herbivore that is coming to eat me as a plant, I'm going to recruit your enemy. If you're coming to eat me, I'm going to recruit your enemy to come and finish you off. And so therefore, that's something I can do as a plant. Plants can also inform their own neighbors and cousins and friends and sisters and brothers about imminent danger, such as the presence of a predator, okay? And then, of course, they can access and respond to local opportunities for future benefit of the individual. All of this has been shown by research. Most importantly, plants do this and much more by sending chemicals, as I told you already, by sending chemicals to communicate with their environment. Neighboring plants sometimes receive, as you can see on the slide, receive the signal, and then they immediately start making changes within themselves that make them more resistant to imminent injury and assault. So all of that is going on as well. And so therefore, plant communication is the most fascinating area in modern science. A lot of us are doing research on it about different aspects. I will come to telling you what little bit of this that we are working on in the lab. But remember always that plants are very carefully considering their environment, speculating on the future, conquering new territories and enemies, and they are often capable of extremely high resolution forethought, okay? what we perceive as fragrant perfume. You all know the perfume, the scent of jasmine, right? And I told you it's a terpene. It's a chemical that is released by plants for communicating with pollinators, with us, with different kinds of potential, friends and foes in the biosphere. So what you perceive as fragrant perfume is actually a sophisticated tool which has been created or evolved by plants to entice pollinators, also to discourage microbes and to fend off various predators. Now, what you see on the slide here are chrysanthemum flowers, which are simultaneously beckoning and combating, I'm so sorry, beckoning and combating this particular butterfly. So this particular butterfly is on the good books of the chrysanthemum. It has followed the aroma of its flowers to a nectar reward. And I hope you all know what nectar is, right? And however, chrysanthemums are also producing a particular kind of toxin, which is called the pyrethrin. It's a volatile insecticide, often used in insecticidal sprays. And this can completely thwart their six-legged herbivores with bad intentions. All right, so the butterfly is happy because it gets nectar and pollinates the plant and helps it reproduce. Whereas those other insects which are biting the caterpillars, they're not very nice, and then they don't have very good intentions for the plant. And so then the pyrethrins are going to be released only then, not for the welcomed guest, the butterfly. Okay, so in all this, I could tell you countless stories, and you could read much more about the kinds of stories of plants and the chemicals that plants produce and how those chemicals help plants. But overall, in general, in the world, as far as we know, there are more than 35,000 unique biochemicals that are produced by plants that are not required for their growth or development. So this is not for their growth and development. It is for the secondary adaptations, okay, for communicating with the rest of the world. And we call them, in general, isoprenoids, okay, because there's a particular pathway in the plant that makes these, and the first product of that pathway is a 5-carbon unit compound called isoprene. So all of the other chemicals that are produced after isoprene are called the isoprenoids because they are coming from isoprene, okay? So these isoprenoids, mostly released as volatiles, constitute the most diverse group of chemicals that plants are making many other chemicals. So that is also something you need to know. But this is the largest group, and this is the group that we are investigating in the lab. The complete complement of isoprenoids. Now, do you know that the complete complement of genetic material inside your cells, inside the millions of your cells, what is it called? It's called the genome, okay? It's your genome. And every single cell of yours has loads and loads and loads, billions of base pairs. And if you stuck all of these base pairs, if you strung them out singly, ek ke baad, ek ke baad, ek ke baad, ek, you would go for your single body and your millions of cells, every piece of DNA arranged end to end in a string would take you from Earth to Pluto and back, okay? That much DNA, that much genetic material you're carrying within yourself all the time. You're a complicated device. But coming back, plants also have more, not just as much as you, more genetic material than you. And all of that genetic material is called the genome of the plant. Similarly, the entire complement of terpenes or chemicals that we are interested in that help plants communicate is called the terpenome of the plant, okay? Billions of tons of volatile terpenes are released or emitted annually by the plant kingdom, mediating very specialized adaptations, as I told you about, mostly defense, mostly communication, but much more. Terpenes also have many industrial uses that humans have found for them. Plants are not making terpenes for us, but we have found, as always, we have found enough to be useful to us all around us, as humans have done since time immemorial. Well, not since time immemorial. Humans are the latest entry into the I would say into the earth. So if you could just stretch your hand fully, you know, and you started from the birth of earth at one side, and the edge of your other end, the tip of your nail that you forgot to clip this week is where humans came into being. All of the other time entirely is other things going on. For these 4.5 billion years since earth was born, human presence has just not even scratched the surface. So still, I should say, from our time, in our time zones, from time immemorial, indeed, we have found uses for things. And so humans have found uses for terpenes in biofuels, feedstocks, fragrance, flavor, the cosmetic industry, the medicinal industry, pharmacology, and all sorts of places. Okay. Coming back to the word that I taught you earlier, variability, the variability in the plant chemistry that we are interested in. It is not, the variability is not just in the types of chemicals that plants can make, or the structure, of the chemicals that plants can make, but the variability is evident at all levels, as you can see potentially in this image. Variability is there between plant species. So different species emit different kinds of chemicals, and different individuals within the same species, within the same plant, the leaf is emitting something else, the flower is emitting something else, the roots are emitting a completely different set of compounds and interacting with the biosphere or the micro biosphere around just that part of them. So here I'm showing that on the left side, you have a plant which has the potential, the gray colored nodes or circles are the potential, let us say, compounds that a plant can make under a certain circumstance X. The middle part will come in. So I don't have a pointer. Anyway, so under a certain time, only some chemicals of the total potential of the plant are going to be emitted. Under certain other circumstances, a completely different set is going to be emitted. So there are differences within and between plants, within and between species, within and between locations, within and between stages, tissues, conditions, and God knows what. So what is it that we are doing? I'm going to talk first about how we capture and measure this variability, because we are interested in the variability. You can't understand language unless you first know the alphabet, right? 26 alphabets in English. कितने व्यंजन हैं बताइए, कौन किसको याद है हिंदी के व्यंजन और हिंदी के स्वर, and what is an antastha व्यंजन and what is a davitva व्यंजन? All right, coming back. Write it in the chat box if you know the answer. But the protein alphabet has 20 amino acids, okay? Similarly, the alphabet of chemistry is something that we want to explore and find out. 35,000 are known, but under what circumstances does which plant make which chemical? And that's what we want to know, okay? So the first part is going to be about, how we capture this variability, as you can see on the slide, and then of course, how we analyze these emissions. And finally, have you ever been to a biodiversity park? Have you ever been to a herbarium? Have you ever been to a botanic garden? I'm going to make sure you do the next time you get an opportunity, and I'll tell you what you could do there. Okay, so how do we get emission data? All right, so let us say conventionally, what you want to do is you want to capture the headspace of the plant. And so you say plants that are emitted as volatiles. What is a headspace? Headspace is a space around my head, stuff that I'm emitting right now. If I'm a plant, please. Okay, good. If you're a leaf, if you're a flower, you will be making different chemicals. How do we capture it? You actually literally cover it in a bag, a poly bag, and tie it up a little bit below the plant's headspace. And after that, you come in, take that take that bag out, bring it to the lab, put it through devices or different kind of machines, which are going to identify the chemicals there. And yes, we fail often because sometimes we find that the air mostly is about your carbon emissions from the garis and the cars that were roaming around the plants at that time, or that were on the road right next to. So that also matters to the plant, because the carbon emissions and your carbon footprint is not just impacting your life, it is also impacting plant life. So anyway, coming back, we do steam distillation with plants to identify their chemicals, gas chromatography, mass spectrophotometry, HPLC, many processes. And very lately, we've been interested in using the xenos. If you've traveled internationally at airports, there are newer ways of how the customs police can actually check if you're carrying a dangerous drug. They just put this little nose, a device, digital device around you, and it's a tiny GCMS machine. It'll just scan you completely for whether there are any chemicals on you. We have used now, Ian Baldwin decided that this would be a good thing for plants. And so he brought it back and he applied it to research. And so we started using the xenos for plants out in the field. And so we know from the xenos that the same plant emits a different set of chemicals in the morning, a different set of chemicals in the afternoon, and a totally different set in the evening, and so on. So that's how we capture it. We collected all of this for the last 100 years. Now, see, obviously, wherever I am in the world, however much I might work day and night, me and my students, there is just no way I can capture the emission or the, I have the capacity to capture all of that which is emitted by every single plant everywhere on the planet. I can't do it. You can't do it. Nobody can do it. But what we can do is to be able to do this analysis on a large enough scale so as to draw statistically significant patterns. And our premise for this is that much of what we seek is already existing in published literature. So we used 100 years of published literature to extract information about known chemicals from plants, different plants. And in this, I got a lot of help from colleagues and friends all over the world. This story is how we extracted and compiled plant emission data from literature. This work began 15 years ago, and we created the essential oil database, which is freely available to any one of you who is interested in wanting to see any of these 150,000 records that we have, spanning 100 years of publications, representing 8,000 compounds and many species. And now we want to have everything from everywhere in the world, and we want open access to data, where Karl Malamud, the president of Public Resource, is helping us. And of course, we built this group of students or interns, mostly undergrad, but also school children. We have a nice 11th standard student who's working with us, Ayush, who's done fantastic work with us because he does great coding, and kids are so much better at coding, I've seen, than most of us who are more senior. So we have the CEV Open Plant Atlas, where we have all of these students are our younger colleagues, and there's Peter Murray Rust at Cambridge and myself here at NIPGR, and we are together, and we are with Emmanuel Faria from Brazil, who's running the Very Clear cosmetic company, and we are extracting information from plants. The next couple of slides are made by our intern student Amreen Hamadani. She explained it so well that basically there's all of science at one place. And all of this science comes into published literature. So there's lots and lots of papers and books that you could read. But could you read all the books in the world? No, you couldn't, right? And so therefore, it's important to find ways of mining such full texts, and that's called text and data mining. Okay, so we are mining these repositories, and that is where we think we can make sense from the publication deluge, which is otherwise impossible for you or me to read everything. Well, those of you who are students already know that when your teachers give you too much homework without realizing that homework is not just in this subject, but other subjects also, you can't read it all, and you can't conclude anything substantial even if you do read it all, and you can't make informed decisions without proper data. This is true for scientists also, and therefore we follow what the great mathematician and librarian from India, Sri S.R. Ranganathan, had said way back in 1933, that knowledge is nobody's monopoly. We should have right to read everything, and we should have right to be able to extract knowledge from published literature. So this is our vision, and we have got many students who are working with us, and I welcome those of you who are even chotas are interested in research. Come and join us as an intern. We have an expanding online mentorship program called CEV Open at the interface of India and Cambridge, run by myself and my close colleague, friend Peter. We call it CEV Open. We have many current interns, and we would be more than happy to have you with us, but you should know how to code, okay, because this is all online. We do some, I'm going to close my story now almost because I see that I have less time. And essentially what we are doing is we are using this research to study the green invasion, or plants that are growing everywhere, and they impact and harm other plants also, because this is a very important issue in biodiversity and ecosystem conservation. We are investigating several invasive plant species, and which are affecting many invaded habitats. We are collecting this data by going out into the field. We are also studying some ecology, but we are mostly interested in chemical ecology and the chemical arsenals that plants are making. We are deciphering the genomes and the terpenomes of these plants. We are using a lot of AI for mapping the invasion features of different plants. And all the time, we are waiting for more students to come and join us and try and give us refreshing ideas. In India, the worst invader is lantana. I think all of you know lantana very well. It's growing all around us all the time. our research is at the interface of botany, chemistry, and geography, because we need to know where all lantana is growing, what its chemistry and toxins are, and how the plant grows, its botany, okay? So this is my student Yuan at Cambridge and his thesis. This is my student Chandler Ramila at, again at Cambridge, her thesis. And in India, it's Abhishek and Renu who are working with me. We've been able to map the invasion history of one or two species. We use world trade networks to help us understand where the species went, how many 100 years ago, and where it might have colonized and invaded in different parts of the world. Finally, on my last slide, I want to be able to say that you must visit herbaria or be able to see if you can, in some way make sense of the plants that you see around you. Today morning, I was at the Sundar Nursery in New Delhi. Well, I'm in New Delhi, and I went to the Sundar Nursery, and we were able to find different kinds of, we played a game where we were matching trees to the leaves that we had around us. And there's a lot that you can do. If you go to a herbarium, there's a lot of pressed plant potential, because at the herbaria at Cambridge and the herbarium at Kew Gardens, we are able to use plant samples and try and understand what were the genes in these older plant samples. In India, there is far, far, far more scope to be studying herbaria and botanical garden specimen, because we have the biodiversity hotspots like very few other places in the world have, right? So I think, therefore, it's very, very important that all of you become a little more interested or a little more curious about plants, or rather than being plant blind, as most of us are. If I show you a big garden and a monkey sitting there, you will notice the monkey, but you will possibly not notice the meadow or the garden, and that's called plant blindness, okay? So in the, what is the message that I want to be able to leave with you? Well, the message I want to be able to leave with you is that biodiversity parks are a very, very, very important part of our day-to-day life. And I think we ought to, especially after the pandemic, when we've been locked indoors and we don't have access to nature and to be able to see different things. I think it's time for us to go into the gardens and see what we have, and the kind of interactions, note the plant pollinator and bee interactions, the seed disperser networks, and so on. Make records of them, talk to a scientist, you might find it interesting. Recent years have witnessed a huge surge in genomicists coming to work with the herbarium specimen and providing an additional window of research. Looking at living plants in herbarium specimen, even fossils, often gives you a snapshot of time, and that snapshot too is dependent on the preservation and conditions at that time of collection. One of the principal advantages of doing genetic analysis is the addition of this time window, Purane herbarium specimen, what was there at that time, what is there today, and so on. And this effort, gives you a spatial breadth of data, in spatial and temporal breadth of data, okay? So with that, I think I'm going to basically just say that in the future, we'd like to bridge the glaring invasion genomics divide and explore if invasive species terpenomes are enriched for certain specific unique terpenes. And in the end, is that my last slide? Yes, I would only like to reiterate that the dazzling variation that you see in plant chemistry all around yourself is a primary mediator of all trophic interactions like herbivory, predation, parasitism, disease. And at the same time, such interactions feed back to influence the spatial and temporal variation of chemistry within the plants. We are investing this spatial, shifting, temporal mosaic of chemicals, and we are very, very happy to talk about it and to get more of you to be interested. If you have questions, if there's something that you don't understand, please feel free to ask me now or email me later, or join me on Twitter. My Twitter ID is JulianV. And of course, we hope to identify patterns that enable us to understand more about the complex interplay between trophic interactions, nutrient dynamics, and so on. Many of you are very young and are thinking about what you might do with your life later. I would like to just say, think about pursuing science a little more carefully than dropping it, okay? All the very best to you. I'd like to end my talk now. Thank you. Okay, what do I do?
Thank you, Dr. Yadav, for the wonderful talk. And I have some interesting questions lined up here. So Yeah, so someone asks here, could you repeat the chemical of pine trees? I think they want you to talk about the phytochemicals from pine trees that you were mentioning initially in the talk.
Yes, indeed. Have you been to the Himalayan foothills? If you've been to the Himalayan foothills, have you noted that there's a bluish tinge in the forest the moment you, cross, let's say, Rishikesh or the area just beyond. I think it's before Rishikesh that you start seeing that bluish tinge, and we call it the Himalayan tinge, generally, but it's the mountains, and the mountains have a lot of pine trees, and pine trees are making a lot of chemicals. One of the common chemicals are monoterpenes. Among these monoterpenes are very tiny, small-sized isoprenoids, which give that particular familiar tinge to the air around the mountains, in the mountains around the pines. Does that answer your question?
Yes, so moving on to the next question, like you said that plants have a way of judging or in their environment and all. So there's a question here, do plants communicate in response to climate change or other natural disasters?
Plants are plants are communicating all the time, but in response to natural disasters, they change their communication very often. In response to everything, plants are changing their communication. So it's basically, I don't want to say they are changing their communication, they're producing a different kind of chemical, which is more responsive to that particular instant and the situation and the condition in which the plant has been put. And so therefore the answer to your question is yes, constantly. Plants are constantly evolving, constantly changing, constantly adapting to the environment as they find it. What we find, for instance, is within a cosmopolitan city like Delhi, the plants that are growing around your highways where most of the traffic runs, and the plants that are more inside the campus. Now, I'm in the GNU campus. Many of you would be in the IIT campus, or maybe you could, depending on where you're living, how far you are from the highways where most of the traffic is. Plants have a completely different kind of growth between the two places. The same species would be totally different in how it's responding to that, and so what kind of chemicals it is producing in both places. So yes, constantly evolving, constantly adapting. They came in long before us, of course, right? I can't hear you. Namrata, is there another question?
I'm not.
I can't hear you, Namrata.
Namrata, you need to unmute yourself.
Should I look at the chat box myself? Arey wah, Ranjana, nitrogen, good job. Absolutely, Aswari, good answer. Vrinda, all of you are pretty smart. Very nice. Okay.
So I think Namrata seems to be having some
think
trouble with her internet.
yes.
And I'm going to try and get the questions from the back end, Dr. Gitanjali, in the meantime, while we wait. I think you have some messages in the chat box which you've been reading.
Yes.
Okay. So I'm not much of a science person, Dr. Gitanjali, so do forgive me. But I'm going to try and take some questions, and do pardon me if I make a mistake somewhere. So there is a question, can class 10 students also apply for an internship? Well, I'm not sure if that's the appropriate question for Dr. Gitanjali. We can, of course.
It's all right. I'm happy to take it because I've been emphasizing so much about interns. We've got a couple of very good interns in our program who are very young, like class 11 students. I've never had a class 10 student before, but I would be more than happy. I know that your teachers would tell you to please pay attention to your board exams, my dear Adhi. do that. But if you have time after your board exams, do write to me. But remember always that this is the digital time, the pandemic. You cannot come to NIPGR or my lab in the foreseeable future. We have restrictions, but you're more than welcome to join me online. And if you join me online, obviously you cannot do experiments in the lab. So what is it that you can do? You can help us digitally with data science. For that, you need to know some bit of coding. So if you do think you have some coding experience and you can do data science with us, more than welcome. Age is not at a bar with us. Most welcome.
Thank you for taking that, Dr. Gitanjali. But I think I have a more relevant question, which is how can we qualitatively identify the plant emissions while visiting a park?
Okay, I don't think you can identify qualitatively plant emissions while visiting a park because plants are never emitting chemicals individually. What plants actually emit is a bouquet of chemicals. Never one, never two. A single plant or a single tissue can also be emitting a huge bouquet of chemicals, usually 100 to 150 chemicals at a time. And imagine in a park, 10 different trees or 100 different trees or shrubs or even grasses emitting completely different hundreds and hundreds of chemicals. Look at the volume of data that you already have. So separating it manually out of the question, using AI to do this, very good. But even so, identifying it first is a question. So that is where I told we are using different methods like gas chromatography. You need to be in a lab. You need to have these high-end devices to be able to identify these chemicals that are being emitted. So you need to capture the space, the head space around the plant, bring it back to the lab, or else you could take the devices straight to the park, put the device there overnight. Some of our students at Cambridge are doing this for in parks and gardens with nectar. So they want to understand nectar chemistry, also the chemistry of certain blossoms. And so they leave their, what should I call them? So we leave these capturing bags out there. Like some of you might have played with butterfly capturing devices, small little nets that you have. For chemicals, you can't have a net because it's permeable. So we have different kinds of bags, but we collect this chemistry and then we bring it to the lab and then we use different chemical methods. And so we first identify this, which is qualitative, and then we quantify how much of each chemical is being emitted in a given bouquet at a given time. So the qualitative as well as quantitative data together is extremely important for our work. And yeah, so do we do it in the lab? When things get better and times get better, then I would be more than happy to welcome you to the lab sometime as students and show you what kind of work we do in chemical ecology. Thank you.
Yeah. So Namrata is back. I think you had some internet trouble there, Namrata. So I just filled in for you. So over to you again. I'll hand over back to you.
Yeah, so there's an interesting question here. The students are asking if there are home experiments that they can do to learn more about plant communication. Is there something that they can do at home using DIY materials? Yeah.
Oh, lovely. I would say there are some very interesting things that you can do, but rather than saying do it at home, when you say home, if you imply indoors, I would not be very happy to ask you to do experiments indoors because plants are natural. They need to be out there in the open. And so think about doing it in your garden. Think about doing it in the neighborhood park. Think about going to a biodiversity park. If you're based out in Delhi, think about going to the Sundar Nursery like you can. And there, go once a week, wherever, choose a place that you would do it, even if it's your own garden. Start recording data every weekend, let's say Saturdays and Sundays. Sit down and try to see, first of all, how many different plants do you see around you? Let's say you capture about 10 or 15 different shrubs, maybe a couple of trees. Record them in a notebook. That will teach you how to do scientific record keeping. And then you make a note of, what are the different insects or birds that are visiting these plants? Birds usually, when they visit a tree or a plant, it is as a seed disperser. They are eating the fruit or they're eating the seeds and dispersing those. Insects, when they visit plants, they're pollinating them. So you can create by insects as well as the plants. If you do it consistently for some time, maybe if you continue to do it weekends by weekends for your entire summer and then do it again in the winter, you would have some very interesting data. Now, more often than not, what happens is when you're doing this kind of an experiment, the biggest issue is taxonomy. You don't know who is that poor plant and who is that poor insect that visited. You know the bee, but you wouldn't know that there are 4 kinds of bees. Did you know that there are 90% of the bees are solitary bees? They live alone and they nest alone. And we always read in books and we've grown up conventionally thinking that bees have this huge hive and they have a queen bee and they have soldier bee. All that is true, but 90% bee species are solitary. But anyway, so you can't recognize those bees. What you can do is pick up your phone, if you have one, start clicking pictures. After a while, you will recognize that the same insects or the same birds come to the same trees because they particularly prefer these for visiting. And if you make these records, like if this was B1 or B2 or butterfly 2 or butterfly 4, and this is the picture of that one, this work could be extremely interesting and extremely useful for a lot of researchers. And you could join a lab, do that for your internship, join me, and we will build a plant pollinator network, or we'll build a seed disperser network, and we'll see how it changes through seasons, and we will understand what communication is happening out there. And this is something you can just do at home. Does that answer your question?
That answers the question. And so there's a follow-up question I found for, like we were discussing, that plants would release phytochemicals in response to different environmental conditions, whether it's natural disasters or some kind of stress. So does the quantity change drastically? The quantity of phytochemicals that are being released, do they change drastically?
I'm thinking of what kind of an example should I give you? All right. So there are certain, if you visit a jasmine shrub, or if you've got the tulsi plant at your home, most Indians have them at home. Your mama or grandma would be taking leaves of the tulsi for the tea that they're making. Try and smell the tulsi. Tulsi is very, very typical. It's got a very strong smell in the leaf. In the mornings, you'll find a completely different scent, very strong. In the afternoon, slightly lesser, sometimes in the evenings, none at all. But then we have flowers like the Rat Ki Rani, which will only be emitting its chemicals at night, not during the daytime. So things, yes, chemicals changes. I told you, variability is the key word, and the variation in chemistry is the critical factor that plants are using to their advantage. They know that the particular pollinator is only going to visit at night. It's a night owl, it's a bat, it's something else. And so therefore the chemical is going to be emitted only at a certain time, and therefore the time and volumes and amounts vary constantly. We do measure these amounts, and these are very important for us because the signal of whether and what is the communication happening between the plant and the biosphere depends upon not just about what are the chemicals, but how much of each chemical is being produced to form a bouquet, a given bouquet, right? So we do need to know the amounts, and we have devices that can measure these amounts very to a great resolution. And yes, it's true that quite often there are certain compounds, many chemicals, whose changes are so minuscule and so insignificant that you wouldn't be able to, you wouldn't think that that's a great difference, but to the plant and to the pollinator, or to its target who is trying to communicate to, that difference would be critical and very important. So slight differences can also be important. Major differences always do happen, recognizable by ourselves. And humans, I'm telling you, we are on the visible spectrum. There's much less of the light that we see, of all the light that is there in the world, and all of that you cannot see. Just a little bit is the visible spectrum. Similarly, our sense also very much in a tiny spectrum of all the scents you cannot fathom, but others can. Insects can. Does that answer your question?
Yeah. So we are talking about the smell of phytochemicals a lot, the smells we get from these plants, right? So do all phytochemicals have a very distinct smell? And also, do these smells have any therapeutic properties? Can they cure diseases or something?
I don't want to say that a certain scent is therapeutic, but there is a huge industry out there which is called aromatherapy. And it's not a joke. Aromatherapy is, so plants have the ability, or scents of plants have the ability to relax you, because these are literally chemicals, and chemicals can modulate your own way of thinking, because much of what we do, most of the food that we eat also has drugs, right? And those are chemicals too. And and all of life is chemistry, essentially. And so therefore, yes, plant scents and plant chemicals do have the ability to impact human well-being, and this has been used for hundreds of years in many different fields. Of course, in pharmacology, more so in aromatherapy, more lately. But this is nothing new. It's nothing new, and for a lot of plants, it is well known what is that particular chemical that makes the change. But just like in case of genes, there is no, when it was first discovered, the dogma or the idea was one gene, one protein, like one gene makes one protein, so one chemical makes one scent. That's not true. It's always a bouquet, and therefore the recipes can be very, 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 very important. So you will find these days in in your home groups and family groups, WhatsApp messages, I find so many of my senior family members sending information like, is पौधे को सूंघ लो, COVID खत्म हो जाएगा. That will not happen. Hello, that will not happen. इस पौधे को खा लो और cancer चला जाएगा. These are things, these are myths that you need to be aware that yes, plants have the potential to do much more than we can ever understand, but that doesn't mean कि तुम ये पत्ता तोड़ के खा लोगे तो कैंसर खत्म हो जाएगा या कुछ ऐसा drastic हो जाएगा, because again, humans don't understand these combinations in these recipes, because when some strong chemical is being made, there could be equally other negatively strong chemicals, and like hundreds and hundreds of years of sages and people working. I'm not just saying scientists in labs, but even in the ancient scriptures, the sages and the myths who were working with plants, they were not creating plants bahar se, like you've read Asterix, right? So who is he? Getafix goes and he pulls mistletoe out and he mixes it up in something. He mixes it up in something. He has a recipe. If you just plucked the mistletoe, you could think about kissing somebody under it, but you couldn't think about eating it and changing things for yourself, my dear. No. So be careful, because there's a very, very fine line between myth and science, and you could easily topple off that little edge, cutting edge. Do not topple off that. Remember that plants and everything about nature, there is much that humans don't know, but that doesn't entitle us to immediately think about answers like is particular plant say aisa kuch ho jayega. Ho bhi sakta hai, but I don't want to be the person to encourage you to believe that. Please don't.
Thank you for bringing up that point for myth and science. That's something really important. Here's another interesting question. We know that plants are producing phytochemicals in response to herbivores as well, to ward off herbivores who are essentially acting as predators for them, right? So do you think, would you say that we need to reconsider the ethics of vegetarianism when we see how plants behave or react to herbivores?
Interesting question. Wow. I always find questions from students coming completely out of a window where I was not expecting. Vegetarianism and veganism and the fact that plants can harm animals too, and therefore you should not eat them because you don't want to harm animals. Hello, don't go in that direction. I don't think this is I'm not the right person to answer that. A lot of the concept of and one's belief in veganism and vegetarianism arises from your personal feelings. I was recently talking to a friend of mine who turned vegan about 10 years ago, but she became vegetarian like 20 years ago. We were both in school, and it didn't happen because she saw something. It was because a boy in the classroom came and said, "Maine kal ek पता नहीं cockroach मारा था, भगवान जाने क्या किया था, कुछ किया था," and she got so revolted by it, and she said, "Oh no, never will I harm an animal, whatever, however small or little it might be." Plants are constantly harming animals. So just because you are a vegetarian, you can eat plants, don't make it because plants don't harm others. Plants can be extremely wicked. You know about this particular, what was the case? So there's this, oh gosh, I'm not the right, I wouldn't tell you right now. So what happens is, and this is really gory, so those kids, if you have little brothers and sisters, just send them away right now because this is going to be so bad. So there's this particular caterpillar that comes and eats the plant, and this plant doesn't like it, and so it calls a wasp. It emits a certain chemical, and the wasp comes flying and it lays eggs inside the caterpillar. So when the caterpillar grows, all of those eggs burst out of its skin like in a Halloween movie, and it has the worst kind of death that you can imagine, and that death was brought to it by the plant. So if you want to stop being a vegetarian today, don't blame me and don't listen to me, but I think it's all got to do with our beliefs. And so I wouldn't like to, as a scientist, comment on it. This is on YouTube, right? Great. Okay, no more comments.
Moving on to a different question, would you say or would you know of any phytochemicals that are able to do cell repairing or can repair cell damage? Is that one of the properties of phytochemicals, would you say?
My answer would be yes. There are many chemicals that have the ability to repair cell damage, yes. And some of these chemicals are in the food that we eat, and the food that we eat often comes from plants, and therefore, yes, indirectly, this is phytochemicals. I can't offhand name any because I'm not a biomedical researcher, but there are surely chemicals that would be able to do that, and plants would be making these chemicals, at least in some amounts, if not huge.
So yeah, thank you for that answer. And we see plants, like we're talking about plants communicating with both animals and how we are able to assess plants in our environment as well, so humans as well. So like humans keep pets and we interact with them. Like with animals, there's a more nuanced communication between humans and animals. So do you think we'll ever be able to use this information, this phytochemical database information, and develop a more nuanced way of communicating with plants like we do with pets? And can we help them out? Can we help these plants to grow or develop much more better in a better way? Can this interaction be possible?
Well, but the answer is obviously yes. Yes, indeed. You should grow more trees. You should visit more gardens. You should plant more shrubs at your home. If you're indoors and you don't have a garden, try to have a hanging garden on the walls. Try to think about that. If you've got a little park or access to some green spaces, think about making a Miyawaki forest. If you're a student and you've never heard of the Miyawaki forest, read up about it on Google. Do a little project on it. Tell your classmates about it, and you will discover that in cities, most of us, because of course of the pandemic, but even before that, many of us did not have access to real big forests. And so therefore, Miyawaki, a Japanese scientist, came up with a mechanism of building these small forests in cosmopolitan areas, which would use the ecology and make use of the knowledge of the ecology and biodiversity of that particular location. So you should definitely do that. And with the previous question, see, now the more you think about it, the more you come. Taxol, isn't Taxol a chemical that is produced by Taxus baccata that is used for cancer? It is the single most important drug that we know about for cancer. And so therefore, yes, indeed, and it is able to handle a lot of cell division issues. Then there's things like also in case of the roads inside cells, what are the cytoskeleton? The cytoskeleton also has a particular kind of protein that is actin, right? Actin, and there's this particular plant chemical that is able to block actin, and that is also extremely important in that is the main cause of certain diseases, and so plants can help you against that, and so on. Most of our drugs today are plant-based drugs, and so therefore obviously plants are helping us all the time in all of our medical research, indeed. But coming back to the current question, yes, you can do so much with plants. Grow them at home, pay attention to them, grow them outside if you can. Don't let the neighborhood guy electrician come and break it up. Just 2 weeks ago, I was not in India, and our engineering section called up and said, from our cameras that are there built for keeping out thieves and intruders, we find that your big bear cut pear is coming in the way of the camera, and so we are going to cut the bear cut pear. I said, can't you just move the damn camera? Why do you have to cut the tree? It's flowering and blossoming. And then this guy says, oh yes madam, yes, we can do that, but pehle se chamkega nahi unko. So therefore you could make that difference, and you could make sure that people don't cut up trees around you, and let them live, and let them have they can't speak for themselves. You can speak for them like you do for your pets. Please speak up for trees particularly. Okay, all the best. Thank you. I hope that answered your question.
Yeah, thank you for that. And can you elaborate a little bit on how exactly scientists identify what chemicals these plants are releasing, and elaborate on the GCMS that you were talking about?
We are doing this in the lab almost all the time. So plants are emitting chemicals at all times. So what we do is there are two ways. One is that you bring the plant to the lab, or you grow it in the lab only, and then you put it in a little glass box. If you're growing a single plant in a little pot, you could grow it in something called the plant growth chamber. We have these chambers, and these are very expensive spaces where you control the light, you control the air, you control the kinds of gases that the plant has access to, and so on. The temperature, the humidity, everything in that plant growth chamber is controlled. We grow the plant there. And when it's time to understand what was the chemical it was making, or let's say under a certain disease, so we provide a certain, some kind of a stress to it. Drought could be there. We want to understand everything about crops, right? So we provide drought to the plant, or we provide some kind of a biotic stress like a virus to the plant. And then we then chop it up, put it into boiling water. That's called steam distillation. The steam from that water comes out, and we capture it all along in a little glass tube, and then we put that tube through a machine called the gas chromatography machine, which lets you see in that tube, in the gas that came out from, in the headspace that came out from the chopped up plant, what different chemicals were there. And each chemical is visible as a peak. And you identify these different peaks based on the molecular weight of the particular chemical. So each chemical has a unique fingerprint. It's called a chemical fingerprint, okay? So the gas chromatograph is like a paper map with these peaks, a plot with the peaks, and we identify these peaks based on many different dictionaries. The dictionaries are actually called databases, and then we match the peaks to those databases, and we say, okay, this was the chemical, and it was emitted or produced in this amount in that particular plant. But more often than not, you can't really chop up every plant and then find out what it's doing, right? You can do it if you're studying a particular disease or a particular stress, and if it's just one plant. But suppose there's a forest, and you want to know what's happening in, let's say, Chakrata as compared to Devban, as compared to Kurseong or Darjeeling, right? So you can't just chop up everything, and your lab doesn't have the space. So then you go there and you capture the headspace of particular plants, and you should always do replicates. There is a scientific method to things. Different populations, different individuals, capture the headspace, bring the headspace back to the lab. Capturing the headspace itself has many different methods of capturing the headspace. You could put a little polythene if you're a really, you're just yourself. You have nothing, no access to labs, no access to big devices. Take a polythene, cover it up, close the polythene carefully, bring it to the lab, give it to me, and we'll try to work it out. Or else, if you're rich, like some of us are, you take these little pen-sized devices. These are injections, and you do still need a polybag light of like a space that is closed enough. So, and you have to leave that with the plant overnight so that whatever it emits and makes would be remaining within that. And then you inject that little injection into the polybags, and then it'll drink it all up, and you suck up all of the air in there. And when you suck up all of that air, everything is in this vial. So then you create these vials, and you bring those vials back, and this allows you not to carry 50 different polybags to the lab, but 50 tiny vials. And these vials can be put into devices that will keep them absolutely safe while you're coming back from the Himalayas, while what if there's a storm or something, and you bring them back to the lab, and then you again insert them into these machines called the SPMEs, or all sorts of HPLC machines can take it also. And then you mix it with a kind of buffer, or you could put different kinds of solvents, and then again you get these peaks which let you see which is the chemical, and both can be measured. Have I gone a bit beyond, students, or has that been sensible enough for you to understand? Please stop me if you think I'm using terminology that you don't get.
Whatever it is, Dr. Gitanjali, it's very, very interesting, but I hate to play spoil sport here. We will be running out of time soon, so I'm going to interrupt and say a big thank you to you, Dr. Gitanjali, for this very interesting session. I promise I'm going to be more observant and more communicative with my plants around me. So that's a promise I make today. Thank you, Namrata, for your very valuable time. This was a great session, and I'm sure our audience too enjoyed being a part of the discussion as much as I did. We will be sending a recording of today's session via email to all registrations, and the video will also be available on Ashoka's YouTube channel. I would also request all our participants to take a minute to fill our feedback form so that we can constantly work on delivering excellent lectures like today's lecture. The link to the feedback form is in the chat box and will also be emailed to you. The next and final lecture of Scientifically Speaking Season 3 is Shapes and Geometry of Surfaces, which is scheduled for Tuesday, 9th November 2021 from 6 to 7 p.m. This session will be delivered by Professor Mahan, MJ, Professor of Mathematics, Tata Institute of Fundamental Research, TIFR, Mumbai. So before we wrap up today, I would like to inform our audience that round 1 admissions of Ashoka's undergraduate program will close on 10th November 2021. Please note that you can secure your admission at Ashoka without your 12th board marks. Join us for an open house on the undergraduate program and admissions process on Saturday, October 30th from 11:30 to 1 p.m. This session will be delivered by Ali Imran, Vice President, External Engagement at Ashoka University. The registration link for this is provided in the chat box. With this, we come to the end of today's session. If you have any further questions regarding today's session or would like to know more about Ashoka's unique interdisciplinary science programs, you can visit our website www.ashoka.edu.in or write to us at ugapply@ashoka.edu.in or call us at the number shown on your screen. See you on 30th October at 6 p.m. Take care, stay safe, and thank you once again, Dr. Gitanjali, for this wonderful session.
Thank you very much. I have really enjoyed being part of this. Very rarely do I get so many questions asked, and I'm seeing the Q&A. It's got many more than you did ask. So I'm very, very happy. As a researcher and a scientist and a speaker, there's nothing more rewarding than seeing more questions and people asking questions. Thank you again.
Thank you.
Hello. Namrata?
Yes, I'm here, Dr. Gitanjali. Yes.
Okay, thank you. I was just saying thank you very much. It's been a pleasure to be here today. Thank you. Bye.
Yeah, thank you.